delighted to have with us Professor Dr. Aan Johan Wahyudi, Chairman of Indonesia Marine Biogeochemistry Forum and Senior Researcher at the Research Center of Oceanology, Natural Research and Innovation Agency Indonesia, who will deliver the opening remarks shortly. Today, we are privileged to host two speakers, Dr. Farah Akmal Idrus from the Faculty of Resource Science and Technology, Universiti Malaysia, Sarawak, Malaysia, who will speak on iron and trace element tiny particle with big impacts, and Dr. Rina Zuraida from the Research Center for Climate and Atmosphere, who will present ocean sediment as archive of biogeochemical change. We believe today's discussion will provide valuable insight into the critical role of trace elements and marine sediment in understanding and addressing ocean biogeochemical process and climate-related change. Without further ado, let us begin by inviting Prof. Dr. Aan Johan Wahyudi to deliver his opening remarks. Prof. Aan, the floor is yours.
Thank you very much, Ibu Mega. Selamat pagi. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much for attending this webinar and especially for the invited speaker, Dr. Farah Akmal Indrus and Dr. Rina Zuraida. Thank you for having us here and deliver the topic that's really interesting related to the trace metal and also how sediment have a role in the biogeochemistry cycle in the sea. And thank you very much also for the audience that also joining us today, that we can learn together the biogeochemistry aspect of the trace metal and for the sediment, so that we can have the new insight for our future collaboration. So without prolong my remark, I think we can start the webinar soon. But before that, I would like to I would like to give all of you I'm not sure how in Malaysia we call it in Indonesia a pantun. So berlayar perahu di lautan biru, nampak ikan menari di sela ombak. Selamat belajar di acara yang seru, semoga ilmu di sini bermanfaat banyak. Makan ikan bersama Cici Faramida, cukup sekian dan sampai jumpa. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof. Aan, for insightful opening remarks. Good morning, Dr. Farah.
Yeah, good morning, Mega.
It is the second time we meet.
Yeah. Inshallah, we'll meet again later.
Amin. Now it is my pleasure to introduce our first speaker, Dr. Farah Akmal Idrus from the Faculty of Resource Science and Technology, Universiti Malaysia, Sarawak, Malaysia. Dr. Farah research focus in the distribution, cycling, and environmental significance of trace element in marine systems. Her work shed light on how these tiny particles, though present in minute quantities, play a disproportionately large role in regulating marine productivity, ecosystem health, and even climate-related processes. Today, she will share her presentation entitled Iron and Trace Element, Tiny Particle with Big Impacts, exploring their source, biogeochemical pathway, and broader implication for the marine environment. Dr. Farah, we are honored to have you with us. The screen is yours.
Thank you very much, Mega, for the introductions. I will share my screen. Where is it? Okay, here. Okay. Then you can you see my screen?
Yes, Doctor.
All right, okay.
Mhm.
All right. Assalamualaikum warahmatullahi wabarakatuh and good morning to all. And thank you very much again to Bu Mega for the introduction. And thank you to Prof. Aan for the pantun. But unfortunately, I cannot reply the pantun. I'm not prepared for the pantun. Thank you to Dr. Ida who has invited me to be one of the speaker for today. My name is Farah Akmal Idrus from Universiti Malaysia Sarawak, UNIMAS. So I will talk about iron and trace elements, tiny particles with big impact. For today's presentation, I plan to make it simple and easy to understand, especially for students, young researchers, and those who are still new for the oceanography study. Okay, these are the content of my talk today. Okay, firstly, I will go for the definition. When I read the literatures, there are a lot of definition for iron and other metals, such as trace elements, trace metals, and heavy metals. But some students or some researchers, they're a bit confused what the meaning of this one, when to use it. Okay, so trace elements actually focus are all elements that are available in tiny amount or small amount, trace amount is why called trace. Combination of metal and non-metals, but must be the micronutrients. Trace metals, they are also part of trace elements, but focusing only on metals and also metalloids, and also serve as a important micronutrient. However, for heavy metal, they are metals, but they have heavy there. Heavy means refer to high density or high atomic weight or molar mass. Okay, I give example here. Molar mass of iron and mercury. So iron is trace metals and mercury is heavy metal. Mercury have high molar mass compared to iron. And some heavy metal, they have toxic effect, but not all toxic metals that have heavy by density definition. For example, arsenic. Arsenic is metalloid. It has a toxic effect to the organism. Okay, next we must understand about what are the importance of iron and trace elements in oceanography. So the main reason is because they are essential micronutrient and support the phytoplankton bloom, which control the oceanic productivity. Back then in 1963, Alfred Redfield and his team develop a general concept of macronutrients composition ratio, which called Redfield ratio. It consists carbon, nitrogen, and phosphorus, with the ratio of 106 for carbon, 16 for nitrogen, and phosphorus 1. Phosphorus or phosphate become the limiting factor for the productivity. But certain ocean regions, they have enough phosphorus to fill up the phytoplankton bloom. But certain regions, especially in Southern Ocean, this condition will not happen. And then after that, a lot of experiments, research have been done, and researchers found out that several other trace elements are also vital and important, and the Redfield ratio formula later was extended to become extended Redfield ratio. Here we can see that, at least out example here, the red one are the macronutrients which required in big amount, and then the green one is the trace metals. We can see iron is the highest, has the highest composition unit compared to other trace metals. However, this stoichiometric ratio may vary between phytoplankton species. For example, if you're looking for diatom, you also need to include silicon in this ratio. So iron involved in photosystem 1 and photosystem 2 or photosynthetic systems. And iron also is a crucial element in the regulation of the ocean carbon balance, which can help with the carbon sequestration to the deeper part of the ocean. So here in the table here, I list out some other trace metals and their biogeochemical functions in the marine microorganisms, including in the phytoplankton. Apart from the biochemical functions, some metals such as aluminium, manganese, and lead, they can also be used as tracers. The famous one, for example, aluminium, used for the dust input tracer, and manganese for the benthic supply. Okay, next, we must understand about the concentrations, how much is available in the water, and then how it distribute in the water column. So concentrations depends on the region. So which are the sample that collected. If close to a coastal region, so the concentrations of metals in the seawater usually high, between nanomolar and micromolar, perhaps to millimolar concentration. And then it's become low in the full ocean depth because of many factors we will see later. And then we need to know how it behaves towards the depth. Okay, so I will show after this a depth profile. And then what are the factors that control the distribution of trace metals? So this factor may become the biogeochemical cycle factor as well. Okay, here is the important depth profile for trace elements in the oceans. There are two types. First, recycled elements, also known as nutrient type profiles. They have very low concentration at surface water. Zero here is surface water, and then it's increased slowly towards the depth. And for example, iron, nitrogen, phosphorus, and many other trace elements are listed here. And for scavenged elements such as manganese, they have high concentration at surface water. Usually, they have involved in photochemical processes which convert particulate manganese to dissolved manganese. So that's why dissolved manganese, the surface water are usually high. Aluminium, as I mentioned just now, is a tracer for dust input. If high concentration of aluminium at the surface, usually they receive high dust input as well. Okay, here are the example of concentrations and how it look like. Usually they behave similar with the depth profile here. So I take it from Sunda 2012. This data from North Pacific and North Atlantic Oceans. We can see that this is a micronutrient and the rest is the micronutrient. Okay, next, as I mentioned, we also need to understand what are the factors that control the iron and other trace elements in the ocean. Okay, they're about the same factors. So I take example for iron. So the first factor we must know the input. Okay, the sources where the iron came from. So usually in the oceans, the inputs came from the atmospheric input or aerosol. Atmospheric input also from dry deposition from the dust, for example, and then wet deposition, rainwater, and also snow. So either from the natural phenomenon, for example, dust from the desert, Saharan desert, for example, or from the volcano eruption. So the wind will blow the dust particles into the ocean. And if the location close to the coastal area, and shelf, perhaps they receive iron and other metals from the terrestrial input through river runoff. And then location, if location is nearby, the Antarctica or Arctic, for example, they may receive a release of iron from the iceberg. And when iron enter into the water, so they will involve in the biological processes. We call it recycling process. And then residue from this activity, they may sink to the deeper part of the ocean. And then another source is come from the bottom. Certain region of the ocean, especially at the mid-ocean ridge, they have hydrothermal vent, which can be one of the important sources from the bottom. And so it will increase the concentration at the bottom part. Okay, how we move? They involve with the physical movement, physical oceanography. For example, upwelling will bring the iron or metal from the bottom part to the upper part of the ocean. And then they also have current that help to supply iron horizontally. Okay, I give example here. I took example from GeoTrace's iron data compiled by Conway et al. For four oceans, iron in 4 oceans, we can see, for example, look at the Atlantic part. Iron should behave as this one, very low at surface and then increase toward the end. And then why a certain area here, for example, at this location, Atlantic, they have high concentration at the surface. We must also see the location. Here it seems like near to the terrestrial, near to the coastal. So the depth, the water depth, it seems to be shallow, and they have maybe they have a turbulence that bring along the iron from the sediment into the water. So that's why they have a high concentration at the surface water here. At certain regions, such as here, we can see very striking red mark at the sea floor here. That probably due to the hydrothermal vent. Okay, so similar with this part, Indian Ocean, and yeah. And then for Indian Ocean, for example, have high concentration of surface water and Pacific as well. Here perhaps due to the iron so dust input, because Indian Ocean very close to the there are a lot of desert, but from the Arabic countries, for example. And then here Arctic receive a lot input from the river, especially during the summer. So the melting of the iceberg. So that's why they have patchy high concentration of iron in a certain area. Okay. Next, we must understand about the internal recycling or cycling, and also how iron can be removed from the water column due to the uptake by the biota. So I'll give example here. When iron particles enter the water, so usually they are in the big size, which is we call it particulate iron. And some of it may aggregate and then sink to the deeper part of the ocean, but some of it may involve in the chemical reaction, which change the size of the iron. For example, change to colloidal iron, and then can change to soluble iron. Only soluble iron can be used by the phytoplankton for the biological processes. However, soluble iron in the ocean usually very low, because soluble iron also can easily complex with the ligand and then become a ligand-bound iron. So phytoplankton only can use in the soluble form. So that's why other reactions such as photoreduction, it can break down solid to free iron. Okay, only free the soluble iron is in the form of free iron, iron 3 or iron 2, and they can be used by the phytoplankton here. Okay, next is we'll talk about high nutrient, low chlorophyll region. So remember just now I talked about red-free ratio, which consists carbon, nitrogen, and phosphorus. And phosphorus is the limited nutrient. However, if we saw here, seen here, that phosphorus in the Southern Ocean, especially back then, they have a lot of phosphorus, which enough to be a supply for the phytoplankton bloom. However, the chlorophyll A did not reflect high bloom here at the Southern Ocean. So the questions are, why these nutrients are not being completely utilized by phytoplankton? Okay, so there were several possible causes hypothesized by the researchers previously. One of it is because of lack of dissolved iron. Okay, not about this, the particulate one, but the dissolved part. And then some researchers, they believe that constant zooplankton grazing over phytoplankton may limit the chlorophyll A value or limit the bloom. And some other possible causes, they say that due to the lack of light, because at Southern Ocean, especially during winter, they have 24 hours darkness. And lastly is about the trace metals toxicity. If it happen, it available abundantly. However, after many experiments, researchers agreed that the cause number one, lack of dissolved iron, was the main factor that caused the high nutrient, low chlorophyll to happen. Okay, from this, it have come up with the iron hypothesis. Previously, John Martin, the oceanographer John Martin and his team hypothesized that primary productivity in HNLC regions was limited by the availability of dissolved iron, because iron that have in that region mostly in the solid form. And in 1991, Martin believed that his hypothesis could also reduce the global warming by iron fertilizing the growth of aquatic algae, which is phytoplankton. And then aquatic algae, phytoplankton, is a tiny plant in the sea which will use carbon dioxide, will absorb carbon dioxide, and then it will sequester into the deeper part of the ocean when they die. So in conclusion, for this iron hypothesis, that fertilizing the oceans with iron could result in a phytoplankton bloom, which could absorb carbon dioxide and then sequester it to the deeper part. As a result, it will combat, it will reduce the problem with the climate change. Several iron fertilization experiments in the ocean had been done before, which all experiments show a significant increase in phytoplankton production. However, not all give a good success stories. The several problems occurred. For example, I list out 4 problems here. The first one, the type of phytoplankton that flourish were without shells. So as a result, the plankton bloom was rapidly consumed by the zooplankton. So it will not achieve the carbon sequestration to the deeper part of the ocean. The second problem, certain type of phytoplankton such as dinoflagellate can produce toxin. So for example, a dinoflagellate cause red type bloom in coastal regions. So this toxin can cause the health problem to other aquatic species. The third problem, when large quantities of phytoplankton bloom, so can cause also large quantity of dead phytoplankton. So in the end, they will sink into the deeper part of the ocean. Deeper part of the ocean has low oxygen level. When there are a lot of that phytoplankton, so bacteria will consume oxygen to to decay for the phytoplankton. So so it will cause the deep ocean species to have a problem, maybe jeopardize if they cannot survive in a very low oxygen level. Lastly, about the political conflicts, because ocean has no boundaries. So when a large-scale iron seeding experiment were conducted, which country should give the permit, which country should regulate it and should control or should monitor it. So there's some problems for the iron hypothesis, but I think the good thing with more than the problem here. Okay, so when we want to understand about the influence of trace metals to phytoplankton, we must understand 2 parts, the chemistry part, trace metal chemistry, and also the biological part of the phytoplankton. Okay, we look one by one. Trace metal chemistry. So you must understand about the concentrations, how much concentrations available in the oceans and surface and so on. And then what are the species of the trace metals that phytoplankton required. Okay, for example, we know that phytoplankton or other biological activity require free metals ion uptake. Okay. However, these free metal ions usually easily complex with organic and inorganic ligands. Next, trace metals are involved in redox processes, and then we need to understand which are the stable species, for example, for iron, that will support the bloom of the phytoplankton. And for we see the example here, because the redox depending on the pH, so we can see the redox potential plot with the pH here at pH 8.1, which is pH for the seawater. Iron 2 is available, and also the solid iron and in this form is also available. Okay, next we must understand the biology of the plankton. For example, the growth rate, how much they require for to support the growth, and then biomass analysis, and also which species that we want to fertilize. Is that can we fertilize the toxin phytoplankton species as well? And after that, we can understand how to and try it together. For example, if very low concentration of trace metal available, it can become the nutrient limitation. But it's in the coastal area, if the concentration of trace metal were very high, so it can cause a toxicity effect, for example. Okay, lastly, when you want to analyze for metals, you need to look on certain aspects for the analytical techniques, not only for metal, but for not only for iron, but also for other metals. The first one, you must make sure you do the accurate measurement of iron concentrations and speciation. For example, you're going to analyze for dissolved, particulate, or iron ligands. And then next, you need to choose which analytical techniques that's suitable for your samples and also suitable for if you want to analyze it on the research vessel, for example, you may consider the instrument that can be brought together and do on the research vessel. For example, flow injection analysis with chemiluminescent detector we can detect iron and also manganese if the metal available, so they can glow in the dark and they can give the data there. So this is a stable technique that can be used on the research vessel as well. And then if you can bring back the sample to the lab, you can use spectrophotometry, for example, ICP-MS. And if you want to look on the iron speciation, you can use cathodic stripping voltammetry. And there, I think I give only some examples of analytical techniques. So for other thing that very important is to make sure the clean environment, either during sampling, storage, and also the laboratory. Lab must be, if you want to analyze for iron, especially for very low concentration of iron, a clean lab is a must to minimize contamination and to ensure accurate measurement. Okay, I think that's all from me. Thank you very much for listening.
Okay. Thank you very much, Dr. Farah, for such an informative and thought-provoking presentation. Now let us move on to our second speaker, Dr. Rina Zuraida from the Research Center for Climate and Atmosphere, National Research and Innovation Agency Indonesia. Thank you, Dr. Farah. Maybe we can change to the other speakers. Okay, Dr. Rina. Good morning, Dr. Rina.
Good morning, Mbak Mega ya.
Iya, ya Bu. Dr. Rina mungkin is specialized in marine sedimentology and biogeochemistry with extensive experience in investigating how ocean sediment record and preserve environmental change over time. Her research provides critical evidence for understanding past climate variability, human impacts, and the long-term dynamic of ocean ecosystem. Today, she will present ocean sediment as archive of biogeochemical change, highlighting how these natural records can be used to reconstruct historical process, assess present condition, and anticipate future marine environmental trends. Dr. Rina, we are delighted to have you here. The screen is yours.
Thank you, Mbak Mega. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good morning, everyone. First of all, I would like to thank for this opportunity to present to introduce sediments as a part of the as a record for biogeochemical history from the past. I'm a geologist, so that's why I will it will be a continuation of what Dr. Farah was presented before, that she presented the iron from the water column, I will not only present it iron, but also other proxies for the sediment. So it will be a continuous, I hope it will be a continuous information. But before I share my screen, I would like to apologize that I prepare my presentation in Bahasa Indonesia. Okay, so can you see it now? Can you see my screen?
Yes, Dr. Rina.
Okay, yeah, thank you. So yeah, I changed a bit of the title. I changed it into marine sediments because that is what we usually use the term for the sediments from the seafloor. So we call it marine sediments as archive of biogeochemical change. But Mega has introduced me before, so I will skip it. And the outline is like, I'm sorry, this is in Bahasa Indonesia, but yeah, introduction and sediment formation and proxy for past biogeochemical cycle changes and application of that. So yeah, what is marine sediment? I'm pretty sure that everybody who attend this talk maybe know about marine sediments, but I would just like to refresh it. So marine sediments, it is unconsolidated. When we talk about sediments, it is unconsolidated. So we have to remember that. And then also that the size, the grain size, it varies from sand, silt, clay, and then it also contain not only various materials, various particles. And why this marine sediment is important to study the past changes, either biogeochemical or climate, because it records the climate from actually not just thousands, but also hundreds of years until millions of years. So this is the climate system of the Earth, right? So from land to sea, and then there is evapotranspiration, sorry, from land air and also sea air. So this is the climate system, and all of this system interacted with each other. So the component of sediments particularly in marine environment, composed of heterogeneous sediments, biogenic sediments, autogenic sediments, and volcanogenic sediments. The heterogeneous sediment, it comes from the land here, on land. Biogenic sediment comes from the shell of organisms that live on the sea, in the sea or on coastal area, or it can also be brought transported from the land to the sea. And autogenic sediments, it forms within the sea, within the ocean, like manganese nodule or carbonate. And the formation of autogenic sediment require a very specific environment. The example of autogenic sediment is actually like salt. So like Himalayan salt, they are actually autogenic sediments that was formed in the ocean. Like I cannot remember the Himalayan. I think is it 60 million years ago or something. So when you ingest Himalayan salt, you ingest past salt from the old ancient seas. Okay. And then there is a volcanogenic sediment, which is a particle that is the result of the eruption. So I would like to tie my talk to previous talk that like Dr. Farah used aluminum as indicator for heterogeneous sediment, because we know that the major element that form the rocks, that form the land and also the seafloor is composed of silica and aluminum. So yes, in reconstructing paleoclimate, we also use aluminum. And then there is in volcanogenic sediments, interestingly, for volcanogenic sediments, we use several things because besides aluminum and titanium and silica, which is major element, there is also calcium. While you can see here that in biogenic sediments, most of the shell from organisms are composed of calcium carbonate. So we have to be able to differentiate between calcium carbonate from between calcium from biogenic sediment or biogenic component with the calcium from the volcanogenic sediment. Okay, I try to draw the processes. So this is like a weathering, assuming this is volcano or just a mountain in the tropical region, then we have a lot of rain. We have a lot of chemical weathering instead of physical weathering, because if you remember, physical weathering is usually caused by the what is it? When water freeze during winter, then the rocks will be what is it? Crumbling? No, not crumbling. Pecah, breakdown of breakdown of rocks due to the seasonal, due to the fluid, particularly water that infiltrate the what is it? Yeah, infiltrate the rocks. Okay. So in tropical region, then there are mostly chemical weathering, and then there will be erosion. Erosion usually because of the rain, precipitation, and then from erosion transported either by the river or by surface runoff. And when it enters the sea, it can form delta or barrier island or any type of sedimentary feature. And then when the sediment from the land carries deeper into the deep sea, it will also form lobes, or if there is instability in the slope of the delta, then the sediments will become a turbidity, will be unstable, and they form turbidity current, and then they will be deposit as turbidite deposit on the deep sea. So this is I just want to show you that from source to sink is that for example, Mahakam Delta, which is very famous, of course. This is Meratus Highland, and also there is another highland here, and we can see that the Mahakam River started from far above here. So you can see that there is not much erosion here, but if you see here that the color is lighter green, and also there are some open spaces here, so it must we can be sure that there is an erosion here, and so it transported everything. By everything, I mean that from the rocks, from the source itself, and also from the environment surrounding it, and then bring it to the sea and deposit it first as the lobes in the delta. The lobes is here. This is the delta lobes here, and also deeper into the Makassar Strait. So just reminder, what is it? Short introduction. This is how we take sediment samples from seafloor with research vessels, and then we can use box core or gravity core. For gravity core, we can get as short as 50 centimeter to maybe, usually we use a minimum 2 meter gravity core. And if the vessel is really long, then we can use until 50 meter of the piston core. That is another mechanism for taking samples with a long core. So then we will have a history the changes that we would know over time, the changes over time. So and then while for the tracer or for the water samples, we have to be really clean. We also would like to have a clean environment, but as you can see that we are dealing with mud, okay? So then it means that when we have the gravity core, we just close the end of each section. We usually cut it into 1 meter section, and then we wrap it, we label it, and then wrap them, and then open them on the lab, in the lab, because then we think that the lab will be cleaner. So in the lab, we will do description. This is a type of the kind of description that we do in the lab. Why? Because then we will see this kind of thing. Difference in color difference, and also if you see this is not unimportant, because it means that the sediment is disturbed by organism activity. And when there is an organism activity, it means then there is oxygen within the sediment. So then we will know at least oxygenation level. We will not know quantitatively, but we will know qualitatively. Okay. Now, and then we will subsample each sediment. It depends on what resolution do we need. Like if we do like 10 meters core, we will not be able to do every centimeter. Then we will do like every 10 meter or 10 centimeter or 5 centimeter. And that's what we call subsampling. And then this is the result of the subsampling, and the rest we put in the storage here. So to do the reconstruction, we use proxy. What is a proxy? Proxy is variable that correlate with any variable that we need to do. So like say, we would like to know temperature, past temperature and salinity of the sea surface. Then we will use something that can represent the sea surface water column. So we use the most established, at least until now that I know, is that foraminifera, planktonic foraminifera particularly, because planktonic foraminifera is live on the water column. So that is for the temperature and salinity. And another proxy that we can use is, of course, stable isotope. I will describe it later. Trace element is the same, like what Dr. Farah already talked in previous talk. And sediment geochemistry and microvessel assemblage. So sediment geochemistry, for example, is it can show the redox condition, past redox condition, because it is still very hard for us to reconstruct pH of the past seawater. But then, first we have to know at least the redox condition of in the past. So that's the thing. So and then what kind of material that we can use? We just use this marine sediment from the gravity core. This is, I mean, we just use marine sediment in this talk. But then this is an example of marine sediment from gravity core. Again, like before, it shows different color, which actually grayish color represent marine environment when it is deposited. And this nice color, red usually indicate oxidation, anaerobic, oxidation, but on land, not in a submarine, but it's actually exposed to the air, to the atmosphere. So then it means that it was it was weathering. Actually, this is a weathering of the granite. So before this, before we haven't date yet of this sediment, but we can say that before it becomes marine here, in the past it is exposed to the air. So yeah, so this is land sediment, and then this is marine sediment. And within the marine sediment, we can find foraminifera here, and also ostracod here. This other one I will not discuss. And then what we can know from the marine sediment, we can know sediment composition, texture. Texture is the grain size, and color, like I said, and density, magnetism. That is the physical aspect. And then the biology is this marine sediment will also contain pollen and spores from on land, because they are carried to the sea by the river, or blown by the wind, or also by the runoff. And it can also contain diatom, dinoflagellate, ostracod, and nanofossil, and foraminifera, of course. And the chemical aspect, we can study the element, major and minor element, stable isotope, biomarker, alkanon, biogenic silica, and other things. So I will, because it says here stable isotope. So just a reminder about the stable isotope. The stable isotope is actually element with different neutron number. So we usually do this, right? So this is the element, this is the mass, and then this is the atomic number. But when we write the isotope, we usually just write the element and then the mass of the isotope. So either, or in the text we either use this or this, or the other one is like this. So oxygen isotope, which is in my opinion is very established to study the paleoclimate or paleoceanography or past Earth condition. We use oxygen isotope, and what we use is usually oxygen 18. Why? Because oxygen 18 is actually the most abundant oxygen is oxygen 16. After that is oxygen 18. So we seldom have oxygen 17 is very rare. Okay, and then this is when we say oxygen 18 in the paleo studies, then we do the delta, and the delta represent the oxygen 18, the ratio of oxygen 18 to oxygen 16 samples to the ratio of oxygen 18 to oxygen 16 standard, and -1 and times 1000. So why oxygen 18 is important? Because of this. During non-glacial period, which is now, we are now in the non-glacial period. So the seawater contains oxygen 16 and oxygen 18. But when it is evaporate, then the evaporation is actually carries more oxygen 16. And then the oxygen 16 will be in a runoff. So it will come back to the sea. So it means that the oxygen 18 in the seawater during present condition is relatively low compared to the during glacial, because when it is carried, evaporate, and then it becomes snowing in the during glacial, then it will be accumulated in the glacier. So it will be isotopically heavy in the ocean, and the foraminifera will use that as when they build their shell. So then it means that the foraminifera will also as more isotopically heavy if they live during the glacial period. So this is the foraminifera. This is for the Globigerinoides rubber that is living in the sea surface. So this is their life cycle. So they are actually living between 0 to 50, less than 50 meters below seawater. So that's why we use it as an indicator for the surface sea surface temperature. So this is their cycle. And why we use Globigerinoides rubber? Because it is cosmopolitan, which is we can find it everywhere. And it also live from zero from now, and it's still living now. And we can find it in the sea, and we can also find it in the sedimentary rocks from 800 million years ago. So it is useful for us. So what we study for the rubber, so for the surface water is actually, sometimes we use rubber, but if rubber is not available and there is a saccharifer, then we will use saccharifer, but saccharifer live in specific salinity condition. So then usually then we will try to find rubber. And then there is a global quadrina, and this is Poloniatina. And this is Neoglobal quadrina as well. This is they represent the thermocline. So this is actually what what I have been what I've talked before. So I think my time nearly up here. So yeah, we can reconstruct the paleoclimate. So we will know the glacial interglacial cycle. Why it is important? Because then we will need to model future future climate, and we will need analogs for that, because past climate will be different during the like Eocene, when we are talking about increase in surface temperature of by the most by the worst prediction by the IPCC, then the analog is actually in the Eocene, Paleocene Eocene thermal maximum. And while the distribution of land on the Earth is different than today. And then we can also reconstruct past ocean circulation. And what I think you will be interested in is dynamics of a carbon cycle, because we know that sediment contains a lot of carbon, not content, sorry, menyimpan, conserve, not conserve, yeah, but yeah, they keep the carbon from carbon, so they are one of the carbon sink today. But then there is several things that can happen that will release the carbon to the atmosphere, from the carbon from the sediment to the atmosphere. Such as if you want to read the paper about the landslide in the North Sea that happened, I think, a long time ago. I think I forget. Yeah, but I will try to find it again. So yeah, that's the part of the what we can use marine sediment to study biogeochemistry. So this is just like a carbon cycle. I mean, I'm pretty sure that you all know about this, but this is the part where my part here is in the marine sediment here. As like I said before, that it can be changed by the organism, but the organism can also store the carbon and everything. So this is just a small an example of what reconstruction of what is sediment, the role of sediment as an archive for past changes. We use elemental here. You can see that this is gallium to titanium and barium to aluminum and zircon to aluminum. So we use that to identify product, whether when upwelling happen and then when it not, and so if we use usually calcium titanium as the indicator of terrigenous input. So if the terrigenous input is high, you can see here. Although we know that upwelling during the if terrigenous input is high, it can also increase the nutrient. But in this case, we use barium aluminum as indicator of productivity, and zircon aluminum as indicator of grain size, or zircon rubidium here. Now, you can see here that the grain is upwelling. So from this is 36 centimeter core. We find that when upward, there's a trend upward, that upwelling become intense, and the intensity of the upwelling is actually we can find here from the organism activity. So we try to do this to scan the core that I show the photo before and identify the and measure the size of this organism. So this is actually the same core, but yeah, different scale here. But this is the same core. So you can see that then we use it for the bioturbation index. And in the upper part here, bioturbation index is actually low. So it means that the oxygen in the sediment is low. Why it is low? Because the oxygen has already been used in the water column during the upwelling. So then there is oxygen in the bottom is low during that, because so there is a hypoxia in the surface, in the seafloor. I mean, in the seafloor. So that's why I think it is really interesting. I think I'm happy that even if it is still, I can say simple method like element, we can at least detect that there is changes in upwelling intensity, and then we can correlate it. We can relate it to the organism activity. So yeah, that's all my talk now. Thank you again for your attention. I return it to Mbak Mega.
Oke, terima kasih Dr. Rina. Thank you to both of speaker. Maybe we can now move into the Q&A session. This is your opportunity to engage with our speaker, clarify points, or share your thought related to today's topics. For those who wish to ask questions, please use the raise hand feature if you would like to speak directly, or type your question in the chat box. I will read them out and address them to the respective speaker. Maybe anyone want to ask? Okay. There is a question from the chat. Thank you for the insightful presentation. I have one question for each presenter from Mbak Ida Ihsani. For Dr. Farah, iron is indeed a fascinating micronutrient for marine phytoplankton. To better understand its biological role, could you please explain the mechanism by which phytoplankton take up iron? Is it absorbed directly, or are there specialized pathway involved? For Dr. Rina, how do rivers, such as the Mahakam rivers, influence deep sea sediments in the Makassar Strait at depth greater than 2,000 meters? How can we assess their impact, particularly in relation to heavy metal studies? Maybe for the first time, for Dr. Farah can answer it.
Okay, thank you, Bu Mega. Thank you, Bu Ida, for the question as well. Okay. So how phytoplankton mechanism. I think the mechanism will explain. I'm not really sure the definite mechanism, but from the reading and then say that the uptake from phytoplankton, either they consume it, especially during the growth. We need to understand the life cycle of phytoplankton. So usually trace matter being used directly, so the uptake, they use it for their biochemical processes during the growth rate. So they use it as for the especially for the oxygen, for example, manganese, they use it for, in the photosystem too, as a oxygen producing. So they're splitting the water into the oxygen and iron and other thing. But after that, after the growth rate, so they absorb onto the surface, the particle, the surface. For example, diatom, they have the skeleton, so they absorb onto that one, and so as a suspend on the surface. So I think it can be as directly, it can also indirectly when they don't want to use it, so they indirectly absorb on the skeleton, surface skeleton of the phytoplankton. For example, if they use it, so they need to use for their physiological function. I think I can, that answer your question, Rida?
Terima kasih, Bu Farah. Yes, that's answer my question. Thank you
Thank
very much. Thank you again for your presentation.
Okay, thank you, Dr. Farah. Maybe the next speaker, Dr. Rina.
Yes, thank you, Mbak Mega and Mbak Ida for the question. I tried, not I tried. I will just use the Mahakam River as the, what is it? As an example. And it depends on the sediment load. I mean, not sediment load. Yeah, sediment load from the river, which is it is big river discharge. Sorry, river discharge that carries also sediment. If the river discharge is large, like Mahakam River, Amazon River, and then there is a submarine canyon that what is it that connect the platform. You can from the picture that I show you that Mahakam Delta, there is a shallow platform, and then there is a slope, and then deeper into the Makassar Strait. So then if there is a submarine canyon that connects from the delta, from the Mahakam Delta into the Makassar Strait, then there is a possibility that the sediment can be carried there to the deeper water. That's the first thing that we, the first, what is it, mechanism that can happen. Another mechanism is actually like wind wave and also ocean and current, because I'm pretty sure that Indonesian true flow also affected the Mahakam Delta, so it can also carry it. So if you want to know the heavy metal and you want to be sure where it comes from, then if you want to be really, really sure, and also you would like to know the source, yeah, maybe then you have to take samples from Mahakam Delta. I think there are a lot of studies actually about heavy metal surrounding Mahakam Delta or in the Mahakam Delta. So yeah, you can also compare it to the previous studies in the Mahakam Delta. I hope it answers your question.
Thank you. Terima kasih, Bu Rina. It answered my question. And I may ask you again directly later.
Okay.
Okay. Any question again? There is question in the chat. Good afternoon, Dr. Rina from Valerie Urai. I am Valerie Urai, student from Universiti Malaysia, Sarawak. Thank you for the informative presentation. I would like to ask, do human activities such as Sand dredging at river mouth area can cause disturbance to sedimentation and sediment chemistry. For example, does it affect the carbon sequestration in sediment? For Dr. Farah.
For me or for Dr. Rina?
I think it's for me. Oh.
Yeah. Okay.
Yeah, sorry, Dr. Rina.
Yeah, yeah, that's okay. From what I read, I think there is a picture like the second before last. There's a picture from Antarctic, and they are talking about the effect of anthropogenic activity into the not Antarctic, sorry, Arctic ecosystems. You can read that. It's also, I think, dredging will, of course, dredging will affect sediment. That's for sure, because sediment is deposited in layer, right? So when you dredge, I don't know the height of a dredging bucket. Is it like assuming it is like 20 centimeter, then at least you take 20 centimeter layer, and it depends on the sedimentation rate in that area. Say in the deep sea, like Timor Sea, 1 centimeter represents 60 years. Imagine. But then maybe in Jakarta Bay, I forget 1 centimeter, how many years? Or in Banggai, 1 centimeter is say 10 years or 5 years. So it means then when you dredge and the bucket height is like 20 centimeter, and then the sedimentation rate is 1 centimeter per 1 year, say, then you will take 20 years of sedimentation from that. And 20 years, what happened on land in the water discharge area? What happened there? What changes there? Is there increase in human activity there? Is there or is the other way around, like a decrease of human activity? If it is a decrease in human activity, probably then the ecosystem at present will be better, and before that will be the ecosystem was worse, or if the online area becomes worse, then it's the other way around. So yes, it will change the ecosystem of the seafloor, of course. It will affect the ecosystem. And about the carbon sequestration, it depends what sequester the carbon there. Again, if it is man-made, like you inject the carbon or something else, then of course it will also change that. Whether for better or for worse, then we have to check it first, because I cannot say, but because in my experience, it will depends from areas to area, area to area. I hope it answer the question.
Thank you for Dr. Rina. Maybe Pak Antoni, are you want to ask something? Pak Antoni? Okay. Maybe thank you Valerie Urai for the answer, Dr. Rina. Dr. Rina, maybe I want to ask about the recommend sampling. How about the interval? It is must be we determine seasonal, annual, or decadal for the sediment sampling to accurately capture biogeochemical trend without missing short-term variability. Furthermore, is the analysis limited to foraminifera for the phytoplankton species, or can other indicator be used? And how does this approach apply to the estuarine environment? Dr. Rina.
Yeah, okay, thank you, Mbak Mega. Yeah, for the sediment, like I said, because sediment needs time to settle, right? So then I would suggest sediment for if you want to say like interannual. intra-annual, sorry, intra-annual. If you really want to do that, then you have to take samples from river in front of the estuary or well, when we say estuary, then it is a sedimentary deposition environment that have several characteristics. So I will just say river mouth. So then you will need to find big, I mean like river mouth of large river discharge that has a large river discharge, you know. So then first, then you have to know like what is the depositional the sedimentation rate there. It's so first maybe I don't know you can just put a box on underneath it and then see after maybe on the seafloor and then see a box, you know, the dimension and everything, and then maybe after 1 month you see if the box is filled or not, you know. So at least then you know the sedimentation rate. That is the simpler way to know sedimentation rate. And then afterward, then if after 1 month is not filled, after 1 year is not filled, then you know, then you cannot use the sediment from there for intra-annual or maybe annual or maybe decadal. So you have to know first the river discharge of this area for the estuary. And then the second one is you ask about the method, right? Foraminifera is not phytoplankton. Foraminifera is Animalia protist. So
Oh,
single cell protist. So yeah, for phytoplankton, we I think we seldom use phytoplankton. Oh, we use that the alkanon, but not specific phytoplankton, but we use alkanon or UK37. The thing is alkanon, as far there is a study that said that the sea surface temperature from alkanon always be colder by 2 degrees compared to sea surface temperature from foraminifera. So people who reconstruct sea surface temperature from alkanon will also do foraminifera or compare it to foraminifera. And particularly, so for the equator, actually, some people say, no, it's not possible. It's not suggested to use alkanon for equator, although newer papers say you still use alkanon for that. But I'm not really experienced in alkanon, so I don't know whether you can use alkanon in the coastal area, from coastal area. That's for the phytoplankton. And then the third one is, you have 3 parts. Your question is 3 parts.
it's enough, I think.
Oh, okay. Yeah.
Okay, thank you,
Yeah, I hope
Dr.
it can
Rina.
answer your question.
Okay, thank you very much. Maybe for Prof. Aan Johan Wahyudi can give the question.
Okay, thank you. My question is to Dr. Farah. So, you mentioned on your presentation about the iron fertilization, or in the common term is ocean iron fertilization, that categorized as geoengineering, the technological approach for carbon dioxide removal, which is by adding bioavailable iron to stimulate phytoplankton bloom in iron limited region of the ocean, to enhance the photosynthetic carbon dioxide uptake and eventually increasing the carbon sequestration to the bottom of the sea. My question is, do you have any example or success story of this approach? And then the second is that in Indonesia, OIF may not feasible since it's actually explicitly regulated under the London Protocol 2013 amendment, that only legitimate scientific research may be permitted, but in Indonesia it's maybe strictly not feasible So, and also commercial OIF is not allowed under the protocol. So how about in the your country, in Malaysia? Thank you.
Okay, thank you, Prof. Aan, for the question. I'm actually not sure about the the carbon sequestration by applying iron as a fertilizer in the marine environment in Malaysia. So for because of so far I'm concerned that they probably have it, but maybe not the main concern in Malaysia part. So I'm not sure about the success story about the iron fertilization that help iron sequestration in Malaysia, but I'm sure that in other part of the ocean there are a lot of experiment have done that one. But the good thing is yes, Prof. An said that Indonesia have tried it, so you have experience on doing that one. But in Malaysia, I don't have experience directly doing iron fertilization for the carbon sequestration. So I'm afraid I could not answer directly the question from Prof. An. I'm sorry about that one, Prof.
Okay, thank you very much. Yeah, actually in Indonesia, we don't have any kind of research on the iron fertilization. And also the regulation is too strict in Indonesia regarding the adding or dumping material to the ocean. So that's
Yeah.
why for the iron fertilization is not really feasible to be done in Indonesia. Thank you, Dr. Farah.
Yeah, in Malaysia, so maybe because we don't have ocean, because we usually close. In Malaysia is the surrounding is with a sea, so the sea with coastal water, so usually don't have the problem with the uh productivity problem and so on. So we have a lot of iron coming from the land, and then so maybe that's why the research on the iron fertilization is not the main focus in Malaysia. We don't have any problem about the iron limiting in our sea.
All right. Yeah, thank you very much.
Okay. One question more from Pak Antoni from University Tanjung Pura University for Dr. Farah. Iron is indeed a micronutrient. Does iron or any trace element function in the phytoplankton metabolism? Does iron entering the digestive system inhibit metabolism? Please allow me, Mrs. Farah.
Okay, thank you, Pak Antoni, for the question. Actually, to answer it, iron entering the digestive system does not directly inhibit the metabolism of phytoplankton, because phytoplankton are photosynthetic microorganism. So it's not animal. They don't have the digestive system because it's plant. However, if for digestive system of phytoplankton, so phytoplankton don't have the digestive system, but it involved in metabolic impact such as photosynthetic rates and chlorophyll content and cellular metabolism, something like that.
Okay. Anthony, how about the answer? Maybe any question more about this
Okay,
one?
thank you, Miss Mega. I'm sorry to Miss Farah. What's the metal concentration limit required for the photosynthetic or another metabolic system in the phytoplankton?
To make it limited, you mean?
Concentration of the metal or trace element.
Okay.
For example, iron and calcium and the other element metal. Would you tell me?
Okay, for iron is the concentration in terms of concentration is about nanomolar concentration less than 0.1 nanomolar concentration, something like that for iron, dissolved iron, and for manganese is in picomolar concentration. last time I calculated is around 10 to 40 picomolar concentration for manganese. So that's two main trace metals. So the rest then it will become even lower than that.
Oh, Lord.
Depending, depending on the just want to put an extended refill ratio. It depends how much it require for the phytoplankton. So it's very, very low. Very low. That's why sometimes the earlier researcher they miss it. They miss
Okay.
it because it's only like nanomolar concentration and picomolar concentration very low, but it helped to fill up the bloom.
Okay. Okay, thank you, Miss Farah. Okay, thank you very much. Thank you, Miss Mega.
Okay, thank you for the question, Pak Antoni. Maybe the last question from Yeti Darmayati for Dr. Rina Zuraida. I would like to correlate bacterial abundance and oil pollution history in certain deep sea ecosystem. How should we take a sample in layer from coring sediment? Thank you, Dr. Rina.
Yeah, thank you for the question. So I assume that you already have the sediment core from what I understand there. So it means that you just have to extrude the sediment if it is a bacterial. But I don't know. I mean, like for bacteria, for microbiology, do you need like a really clean environment? How do you, what is it, avoid contamination from areas that it's, yeah, you have to think about it. And then you can just extrude it like what Dr. Ida did. So you push the sample from the core. You just use anything and push it. So you put in vertically, and then you push it from the bottom, and then you slice it every centimeter. So yeah, there is a thing that called core extruder. It's just what is it? Something that has maybe Maida can give you the photo. So yeah, but there is something that called a core extruder that you can use as well to extrude the core. So you just actually push it and every centimeter, and then you slice it and then put it in whatever container that used for the microbiology. Thank you.
Okay. Another question maybe for Dr. Rina again. West Kalimantan has the Kapuas River estuary, so many rivers in our province, but yet research on the aspect you mentioned remains limited. How should we begin to understand the role of this estuary in the biogeochemical cycle of the Natuna Sea, particularly in relation to the impact of its presence? Maybe Dr. Rina can explain more detail for how we can begin the research for the beginner of the status of our waters, maybe Dr. Rina.
Yeah. Thank you. First, thank you for attending this meeting, Dr. Afriansyah. And yeah, maybe we can start doing it. I will make a proposal about that. But actually, before offshore Kapuas Delta, there was what is it, samples that were taken by the Ministry of Energy and Mineral Resources by the Geomarine 3. So the samples are at the Marine Geological Institute cold storage in Cirebon. There are several papers about that. That was a national paper about that. But because we are geologists, so then maybe we can review the paper and take the biogeochemical aspect from that, and then afterward, then we can do a further study from that. Actually, another thing is there was also study. I don't know whether they publish it or not, but there was also I think a collaborative study. Is it? Oh, well, I forget about that. But yeah, there are several study, but mostly by geologists, and then also the PNIAL in the particularly not the Kapuas Delta though, but in the border between Malaysia and Indonesia, and KKP also. But maybe because the study is what is it? Sectoral, segmented. So the information that we have is not really of doesn't what is it nggak menyeluruh gitu loh, apa sih, not holistic. Yeah, but comprehensive. We don't have comprehensive data about that, but yeah, we can start to do it and check all the papers that are related to that, and then afterward then we can do more study about that. Thank you.
Okay. Thank you for the answer, Dr. Rina. And thank you for Mbak Yeti for the question too. Maybe it's enough for the information and for the webinar today. As we come to the end of today's session, I would like to sincerely thank our speaker, Dr. Farah Akmal Idrus and Dr. Rina Zuraida for sharing their valuable knowledge on behalf of the Indonesia Marine Biogeochemistry Forum. We truly appreciate your time and participation in webinar series number 11. We look forward to seeing you again in webinar series number 12, where we will bring you a new and exciting topic to explore together. Until then, please stay safe and healthy. See you next time. Thank you, Dr. Rina, Dr. Farah, and for the participants today. I hope I can meet all of you in the uh, right moment and nice moment. Thank you, Dokter. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Salam.
Terima kasih semuanya.
Terima
See you.
semuanya.
Kasih
Mohon
Bu
maaf
Mega,
jika ada
Bu Rina,
kesalahan. Ya.
Bu
Dan
Farah.
Mega. Terima kasih,
Sehat-sehat
Mbak.
selalu.
Amin.
Thank you, Miss Farah.
Ya, thank you.
Thank
Maybe
you.
nanti saya kontak dengan Bu Mega. Kita boleh
Ah,
kerjasama. Nombornya udah
boleh,
dapat, belum
boleh.
kontak lagi. Saya dah dapat nombor
Siap,
dari Bu Ida.
Siap.
Boleh sekali dengan Pak
Siap.
Anthony.
Ya, banyak yang mau saya tanyakan sebetulnya, Dokter Farah.
Iya, nanti
Cuman
kita
nanti.
bincang kemudian.
Ya, siap, Bu Farah.
Oke, alright.
Mantap ya. Senang banget maksudnya kalau bisa jadi bridging untuk
Penyambung.
kolaborasi.
Insya Allah.
We are happy.
Ya, makasih Mbak Ida.
Ya.
Maaf suaranya
Oke.
agak-agak.
Terima kasih Mbak Mega. You did
Ya.
your best. Ya. Thank you Bu Farah for everything.
Ya, thank you juga.
Bye-bye.
Bye. Assalamualaikum.
Waalaikumsalam. Terima kasih Pak Antoni untuk kehadirannya.
Terima kasih Bu, makasih Bu. Sampai ketemu.
Ya.
Bu.
Saya live duluan ya Pak, Bu.
Iya Bu, sama-sama Bu, kita
Sama
lihat
juga.
Bu.